Hello and welcome everyone. In this podcast, we're going to talk about the fluidity of the membrane. A few other things, but that's really a major theme here, fluidity of membranes. In the last podcast, we talked quite a bit about how when we form this membrane, I'm going to just draw a small version of it here, that because of the nature of the hydrophilic heads, which were shown here in orange, or the hydrophobic tails shown here in blue, that the membrane itself would form a continuous circle with itself, like so, to prevent water from interacting with the hydrophobic portion. So in that sense, the membrane is fairly static. There's not much change with that. However, what we know is that the membrane is anything but static. It's an extremely dynamic structure. There's a lot of movement that happens. And we'll talk about that. There's movement that happens in the membrane, moving side to side within the membrane. And within the hydrophobic portions, we know that these whole lipids can also move. And we also know that we can get some flipping going on here, where these up here can flip this way and these up down here can flip up there. They can do what we call, in a very scientific term, flip-flop. So it's a very dynamic structure. It's often called the fluid mosaic model. Very fluid because there's a considerable amount of movement within the membrane and from one side to the other side of the membrane. Mosaic because within this membrane we have various proteins on one side or the other side. Some kinds of lipids are on one side of the membrane and other kinds are on the opposite side of the membrane. So it's not identical from one side to the other, or from this part of the membrane of, say, the cell or this organelle to this part. So we call it a mosaic. And it turns out that this is very important. So why is this important? Why is it important that we have this fluidity and this mosaic nature of this membrane? Well, we could talk hours, probably days, maybe even years about how important this is. But let's just talk about it in general terms right now. It's important for the function of the cell or of that organelle. The fact that one side looks a little different than the other side allows these lipids to interact with different sets of molecules as opposed to these. So it's very important in the function of that cell or that cellular organelle. It's also extremely important for integrity. The overall fluidity of the membrane, making it more or less fluid, is important to determine the integrity of the entire cell. And how we can change the fluidity is an important component for the integrity of the cell. So in general, we can think of the kinds of movement I just talked about as lateral movement, where the lipids sort of exchange with each other on one side, this lipid here exchange with this one, and so forth. This turns out to be very frequent. In experimental situations, this occurs every second or so. The other kind of movement, which we call flip-flop, where this, say, set of lipids moves over here, and this set of lipids moves over here. This is very infrequent. It's very important, and we'll talk about later on in this podcast why it's important, but it doesn't happen nearly as much. And we don't want it to happen as often, and I'll talk about that in just a second. In experimental situations, this might occur every month. In a physiological situation, it occurs a little bit more frequently than that. But the reason we don't want this to happen too often is because, remember, we said that mosaic feature of this model is that we want these lipids here to look a little different because they interact with different components than these lipids. Now, in the plasma membrane, these lipids interact with the external environment, and these lipids interact with the cytosol. So we don't want a lot of flipping going on here. The lateral movement, on the other hand, is very important that it moves around quite a bit, and it won't impact the overall integrity or function of the cell. And I want to start talking about fluidity factors. The first I want to talk about is a physical feature, and that is temperature. As temperature increases, then fluidity also increases. This could be dangerous because you don't necessarily want the membrane to become too fluid. So how would we prevent that from happening? And we'll come back to that in a little bit. Just like an increase in temperature, we know that if we lower the temperature, then fluidity will also be decreased. And there's a limit to that too. We don't want a complete breakdown in the fluidity in either case, increasing or decreasing the temperature. So how does the cell counteract what temperature can do to the fluidity of the membrane? Both of these have to do with hydrocarbon tail. First is the length of the hydrocarbon tail of these lipids. And the third is the number of double bonds. Or another way of saying that is the degree of saturation. Okay, now let's talk in more detail about how length of the hydrocarbons and the number of double bonds affects membrane fluidity. And to do that, I'm going to draw two membranes here. The top one is going to have fairly long tails, to the point where they're easily interacting with each other, themselves next to each other, but also the ones across the way from them. This one down here, we're going to have small, puny little hydrocarbons. Now, hydrocarbon lengths can vary quite a bit. The range for hydrocarbon lengths is somewhere in the ballpark of, say, 14 to 24 carbons. However, the average is somewhere between 18 and 20 carbons. But the longer the chains are, first of all, you will get more interactions between the tails here. These increased interactions here will decrease fluidity. These shorter chains will result in less or fewer interactions between the hydrocarbon tails, and because of that, it will increase fluidity. Longer chains, less fluidity. Shorter chains, more fluidity. Let's now move on to how the number of double bonds can cause a change in fluidity. Let's first remind ourselves of the difference between saturated versus unsaturated fatty acids. And what we might typically see in a cell are these two hydrocarbon chains. One that is, and I'm going to draw the first one as saturated, like that. Saturated means that every one of these carbons has two hydrogens on them. They're saturated with hydrogens. The other one, and this is actually kind of common in cells, that one is saturated, the other is not saturated. And so again, this is unsaturated. And all that means is that some of these carbons are not saturated with hydrogens. So this one would have two hydrogens on it. But when you get to this one and this one, there's only one hydrogen. And that's because there is a double bond here. And because of that, what happens is it forms this kink in the hydrocarbon tail. And phospholipids like this will have varying degrees of saturation. Sometimes there will be one in there. Sometimes there might be 10 or 15 double bonds in there. And that changes the ability for the membrane to be fluid. So how do double bonds impact fluidity? So let's write down some rules here. More double bonds makes it more difficult for the chains to pack against each other. Another way of saying it is it makes it less flexible, more rigid. This chain, since it has this kink in it, is going to be less likely to interact with this chain, or any chain. This makes this membrane more fluid. Less packing makes it more fluid. Think of your vegetable oil. Your vegetable oil is a liquid. It's also an unsaturated fat. The fat that you get from, say, a steak or from lard, that's saturated. It's a solid. Less interactions makes it more fluid. So let's write the counter to this. Fewer double bonds makes it easier to pack against each other, makes it less fluid. So how can these influence membrane fluidity in temperature, or can they? what can be changed and what can't be changed. So I'm going to erase this and talk about two different strategies. So remember that the relative fluidity should remain stable. Obviously there are some exceptions to that, but that's the goal. The fluidity of our membranes doesn't change that much, but the temperature does, and other factors do. So how do cells account for this change in temperature? I'm going to talk about two broad strategies. The first will be a non-cholesterol strategy. And this is something that organisms like bacteria and yeast employ. Now, bacteria and yeast, they don't make cholesterol, but they do have to respond to a constant changing environment, particularly the temperature. Fluctuations in temperatures affect bacteria and yeast very quickly, so they have to have a mechanism to respond. The other mechanism I want to talk about, I'm sure this will shock you after hearing the first mechanism, but this one's going to be the cholesterol mechanism. And this is something we will see in animals. But first, let's talk about the non-cholesterol way. So I'm going to draw a bacterium here, and here is a yeast cell that's budding. In order to survive, they have to have a way to maintain this relative fluidity. They don't have cholesterol, but what they have done is they've adapted a rather amazing strategy to survive. And what they can do is they can change the length and degree of saturation of their phospholipid hydrocarbon chains. So as an example, when the temperature increases, these cells do two things. First, they make phospholipids with longer hydrocarbon chains, and these chains contain fewer double bonds. So remember, temperature increases overall fluidity. So the cells have had to adapt a way to counter that and decrease membrane fluidity. Longer hydrocarbon chains, they increase the interaction between those chains and thereby decrease membrane fluidity. Likewise, fewer double bonds allows those hydrocarbon chains to stack up on each other with a little more ease and thereby decreasing membrane fluidity. Temperature increases the fluidity, these changes decrease membrane fluidity with the overall goal of maintaining this relative fluidity. And the reverse would also be true. If you decrease temperatures, bacteria and yeast, they would have shorter side chains and more double bonds in an effort to increase the fluidity to, again, maintain this relative balance. Okay, so let's talk about this second mechanism of cholesterol. And again, this is a mechanism used by animals because they make cholesterol. So if we think of our membrane that we've drawn, once or twice here, and we have varying degrees of tails here, and I drew a cholesterol molecule here, not in great detail, but let's think of cholesterol as this small and rigid molecule. So what is this cholesterol molecule doing in the membranes, and how does it affect fluidity? Well, being a small and rigid molecule, it can fit into tight places. There's a lot of gaps in the membrane left over in large part due to the unsaturated fatty acids forming in here that have those kinks. So there's these large gaps, and cholesterol molecules float around and they find these areas. And what they'll do is they'll make the membrane more rigid. They'll also prevent or lessen, is probably a better way of saying it, these hydrocarbon tail interactions. So what's it do as this buffer? Well, we know that as temperature increases, you would predict fluidity to also increase. We would also expect as temperature decreases that the fluidity would also decrease. Cholesterol acts as this buffer and counters this raise in fluidity and decreases it. And at lower temperatures, it increases the fluidity. So for the purpose of this class, I would like you to know that cholesterol serves as this interesting little buffer that decreases fluidity at higher temperatures and increases fluidity at lower temperatures. How it does it isn't as important for right now. But in general, what it does is it helps stabilize some of these interactions to alter the fluidity, the interactions between neighboring side chains. Okay, now I want to move into a different area, and that area is how are membranes made? As cells grow, as cells get damaged, different membranes or new membranes need to replace the old ones. So let's get started with this discussion on how membranes are made. We have the nucleus here, just to give us some landmarks. So we have our mitochondria here. We also now have our ER, which is usually formed around the nucleus and can extend out. And then just to put everything in context, we'll have our Golgi apparatus here. Okay, so let's start with this picture here and talk about how new membranes are made. They're essentially made on the cytoplasmic side of this ER membrane. And remember, what we're looking at here is we have half of the bilayer over here of the lipid bilayer, and we have the other half on this side. And so what's happening here is we have free fatty acids in the cytoplasm that are incorporated into the membrane here. So what I want to do is expand this section of the membrane over here. And up here will be the cytoplasm, and here will be the ER lumen. And I'm going to draw some new fatty acids coming along here that are in the cytoplasm, and now they're going to become incorporated into this membrane. And though I didn't draw it, remember this membrane that was already there continues all the way around. So it wedges this new fatty acid here, right here in the middle. Now we know that when we look at any given membrane, there's not more phospholipids on the outside, the cytoplasmic face of the membrane. We know that they're fairly well evenly distributed, but this mechanism here only puts them on the cytoplasmic face. Alright, I adjusted the drawing a little bit. Instead of 3, I put 4 coming in here because it'll make this next step easier to explain. Okay, so what happens then is, because we want to make sure both sides of this membrane, in this example here, get 2 new fatty acids. And so what happens is there's this enzyme, and it's got a great name. It's called scramblase. And what scramblase does, as you might guess, is it scrambles the composition of the membranes so that these 2 new brown ones here now can end up on opposite sides of the membrane. In doing this on the ER, you end up with a membrane that is fairly even, the same one side versus the other. So as new fatty acids are incorporated in here, they get scrambled around some so that we end up with a fairly even distribution of lipids on the membrane here. However, we know, if we go back to this drawing here, that these membranes will get pinched off here and form a vesicle and get transferred here. And now on the Golgi, we will have some of the ER membrane. And at some point, this part's going to get perhaps pinched off, but it forms a new vesicle. Now this vesicle, it may have a destination to some part in the cell, or it may have a destination to the plasma membrane. But wherever it ends up, and let's just say it ends up here, this membrane that was made up in the ER here, that has a fairly even distribution of lipids on either side, we know that the lipids on the cytoplasmic face and the lipids on the external face of the cell are different. So how can we make it, this membrane, switch around some so that the 2 sides are different? Now I didn't draw it here, but let's just say we have a mixture of glycolipids here. And glycolipids are important to be on the outside of the cell. How do we get these all on the same side? Well, what happens is another enzyme comes along that also has a pretty clever name called flipase. And what flipase does is it flips the lipids so that certain groups of lipids are all on the same side. So what might happen here is during this flipping process, we'll end up with all the glycolipids that are needed to be on the outside of the cell to one side of the membrane, so that as it's going through this Golgi and it hits this vesicle and ultimately ends up on the plasma membrane, that all of these lipids are glycolipids, or the ones that need to be glycolipids are all out there. And we don't have any glycolipids on this side or on this side here. So two enzymes that are important. Scramblase helps to make sure as the membranes grow that they are even. Flippase is important in making sure that the right membrane lipids are on the correct side, so that whether it goes to the plasma membrane or some other organelle in the cell, that the membrane lipids have the right composition on the right side of the membrane. Okay, that's all I have for these two podcasts on lipid membranes. In the first podcast, we talked more about the structure of the lipid, the components of the lipid, the different types of membrane lipids that could form, the differences between bacteria, archaea, and eukaryotes as it concerned the structure and components of the membranes. And then in this second podcast, we focused more on the fluidity of the membrane, how we can maintain certain constant levels of fluidity, the factors that are involved in changing fluidity, and then we talked about how new membranes were made. If you have any questions at all as you're preparing for class or the test, please make sure you come see me. If not, I'll see you later.